Today's topic is exogenous antigen presentation. So, what is basic idea about exogenous antigen presentation? Exogenous antigen presentation is a process where the antigen presenting cell, the professional antigen presenting cell, notice a antigen outside the cell, not inside the cell, not inside its own cell, outside the cell means in tissue or the cell. If a antigen enter in our body and it is noticed by the professional antigen presenting cell, that is specifically the dendritic cell or the macrophages. Okay, so presence of that kind of outside antigen is known as exogenous antigen presence. Okay, so let's move forward. See, so the exogenous antigen presence is solely done by the MHC-II molecule. Okay, MHC-II molecule. The MHC-II molecule is only present in antigen presenting cell. So how it is done? So first, an antigen is internalized by presenting cell. Okay, by any process, endocytosis. Okay, by endocytosis, the antigen is internalized by the antigen presenting cell. Let's suppose a dendritic cell is exposed, a antigen is exposed to a dendritic cell. Now the dendritic cell engulf or by the process of endocytosis internalize the exoantigen and internalize the endoantigen and and process it. Here process means breakdown of that antigen into smaller peptides. Okay, antigen is mainly proteins, so these proteins are break down into peptides of 8 to 13 amino acids. Now it is presented by now is this processed peptides are bind to MHC-II antigen binding cleft, and now the MHC-II molecules uh, migrate to the membrane, and the antigen is presented to the T cells. Now the endolytic pathway degrade the antigen into smaller peptides. So it is a hydrolytic acid attack, which break the protein antigen into smaller peptides. It involves mainly three acidic pathways. The first pathway is early endosome, the second pathway is endolysosome, and the third pathway is lysosome. Now, by these three acidic pathway, the proteins are broken down into oligopeptides. These oligopeptides binds to MHC-II binding cleft, MHC-II antigen binding cleft, and into the get attached into the MHC-II molecule, and then it is presented outside of the dendritic or antigen presenting cell. Now we'll discuss about the actual mechanism how this antigen presenting cell process this antigen and present this antigen to the T cell receptors of CD4 plus cells. So the mechanism involves from the formation of MHC-II molecule. The MHC-II molecules are synthesized in uh, rough endoplasmic reticulum. Mainly, the alpha and beta chain are synthesized in rough endoplasmic reticulum. During the synthesis, a heterotrimer is formed. A heterotrimer of alpha, beta, and invariant chain formed in RR. During maturation, the invariant chain are slightly degraded. Okay, and then delivered to the and this heterotrimer is then delivered to the MHC-II compartment. The MHC-II compartment have certain enzymes which degrade the invariant chain further and convert it into a smaller molecule known as clip. Clip is nothing but class 2 invariant chain peptide, which is actually bind to the MHC-II antigen binding cleft. In inhibiting the binding of antigenic peptide, it remain in that place. So a, a molecule or a molecular chaperone known as HLA-DM. Okay, HLA-DM replace the clip with antigenic peptide. Means the HLA-DM removes the clip and replace that clip with antigenic peptide which is processed. Okay, now the peptide MHC-II complex, now a complex form which is known as peptide MHC-II complex, means it means a complex of MHC-II and the antigenic peptide move to membrane and presented to the T cell receptor of CD4 plus cells. CD4 plus cells means the T helper cells. So the MHC-II present the antigenic peptide to T helper cells. We already know that MSC-II always present the antigen to the T helper cell, and MSC-I, which is present in all nucleated cells, present the antigen to cytotoxic T cell. Thank you.